The overall goals of these procedures are to automate the observation of bee choice behavior at artificial flowers. This is accomplished by first gluing an identification tag on all bees inside a colony with plastic number tags or RFID tags, depending on the protocol. The second step is to set up motion-sensitive camcorders or the RFID readers in the testing space. Next, the artificial flowers are placed in the testing space. The final step is to give the bee colonies access to the testing space. Ultimately, a large database of records is generated, consisting of the time and location of each and every floral visit.
The main advantage of these techniques over other methods, such as recording observations manually in real time, is that these techniques enable us to study the behavior of bees that have never yet experienced rewards on flowers. Bees take time to discover flowers, sometimes even days. Our methods allow us to capture these very rare events.
We can record the activities of an entire colony of bumblebees continuously over an extended period of time and with high precision. This can help us answer key questions in insect behavior about the origins of individual differences.
The testing environment for this protocol must be an isolated room or a 2 cubic meter metal screen flight cage. On the flight cage, a small hole with the diameter of a nickel, or about 2 centimeters, serves as the entry and exit point for the bees. Bee colonies are connected to the test space using a conduit that is wide enough to let several bees circulate simultaneously.